The standards are diluted according to the kit instructions. In buffer containing bovine serum albumin. Doubling dilution is made of the standard. The gel is left for about 15 minutes uncovered and checked to make sure that there's no condensation in the wells before applying the patient serum and dilutions of standard and controls to the wells. Small volume, 3 microliters of each standard dilution is then applied to the standard wells. Being very careful not to touch the edge of the wells or distort them in any way with your pipette tip, because that will affect the results later on. The agarose gel contains a concentration of antibody to human factor H. Now we're going to add patient serum samples and a control to the wells. And if there's factor H present, the protein, factor H protein, will diffuse out into the gel over the next 72 hours. When the factor H reaches, as it diffuses out, when it reaches a concentration that's optimal, it will precipitate out into the gel. It will precipitate out of solution and into the gel as it binds with the anti-factor H in the gel. This will form a ring of precipitation around the well in the gel at a distance where the optimal concentration occurs. So the further away that precipitation ring is from the well, the more concentrated the factor H is in the patient's serum. So the diameter of the precipitation ring is directly proportional to the concentration in the patient's sera. Once you've pipetted the patient's sera, an airtight lid is placed on the gel to prevent the gel drying out, and to allow the patient's sera to diffuse into the gel without evaporation and the gels drying out. So the gel is put in a sealed pouch and then kept at room temperature in a box to prevent disturbance and left for 72 hours.